I'll introduce Dr. Lu Huang. So Lu obtained his bachelor degrees in bioinformatics from Zhejiang University, and then did his PhD in Cornell. After that, he stayed in Cornell, did his postdoc with David Russell, work on macrophages and MTB infection. In 2020, he moved to University of Arkansas Medical Science in the Department of Microbiology and Immunology to set up his own lab. I first met Lu several years ago. We were on the same shuttle to Keystone conference, and I know his work, and I was very much impressed with his work, and I was very happy to see him on the shuttle. So we start to know each other. And over the years, he has done very great work and beautiful work on macrophage metabolism and in MTB infection. So without further ado, Lu, stage is yours.
Sure. All right. Can everybody hear me okay? All right. Okay.
Yes.
So, yes. Okay. So thank you, Jay, for the very generous introductions. And it's also my great pleasure to be here as a speaker for the Friday seminars for our associations. So I will take this great opportunity to meet old friends and also want to meet new friends from all over the places. And also it's a great opportunity for me as a junior faculty to share what we have done during the past 3 years. So I was just talking to Jay and other people that I started my lab right before the pandemic hits. And so it seems it's quite different. But we hope to share the stories and get some feedback from everybody and to form some new collaborations in different areas. So today I'm gonna focus on talking about a lung disease, which is a Mycobacterium tuberculosis infection and cause disease called tuberculosis. And then lots of people may still confuse why nowadays TB is still a problem. And this at least was many people think, oh, TB, we already have antibiotics, we already have vaccines, but the thing is not what we thought. So this is a news website. We found back in a few years ago that TB is one of the most deadly infectious disease worldwide. And of course, if you have HIV, and then it'll make things even worse. And then things when the pandemic COVID-19 hits, this actually make TB make the battle against TB is actually even worse. I think in 2020 we have first time in over 10 years, we have increased TB deaths worldwide. And then according to WHO, people died of TB each year would be around 1.5 million to 2 million. So it's still a quite health problems worldwide. So this is a brief figures just to talk about the life cycle about this intracellular bacteria. So this bacteria is highly contagious. So usually you can cause disease if you inhale as low as one single bacteria can cause infections. So the first, it's well known right now that the first cell types that when bacteria enter the lung, it's a very specialized macrophage resident in the lung called alveolar macrophages. And so once the bacteria actually get phagocytosed by this type of macrophages, and it will initiate a downstream cascade of different kind of immune responses, and then it recruit many different type of both innate and adaptive immune cells recruited to the lungs. So eventually, this bacterial infections cause this kind of structure. It's a hallmark of these diseases, and this is called a granuloma. So the structure, as you show here, this is a mouse lung that infected this TB that forms a granuloma. So as you can see here, the center is highly fibrotic. This is the blue. This is blue indicates the collagen staining. So there are many necrotic macrophages and neutrophils lives in the center of this granuloma, and then on the edge here, there are a lot of foamy macrophages. That means they have macrophages have lots of lipids accumulate inside of cytoplasma, and the lymphocytes like T cells, it's also form called a lymphatic cuff. So a cuff, so it's formed the edge of the granuloma structures. To work with TB, since it's a highly contagious, so it requires special facilities. It's a biosafety level 3 lab. So this is what we have done up, have a pepper respirators to prevent infection happen to ourselves. So this is actually me and my technician when he first certified and passed all the training to start working in the BSS3. So this kind of pictures. And then, so what do we know about protective immunity against this bacteria? So we actually know quite a lot about this infections. So this is a figure from, I steal from this review a long time, almost 20 years ago. So people knows that what are important components from immune system that control the infection. So for example, if you remove T cells, you get almost uncontrolled bacterial growth. And however, if you remove these two components, gamma interferon or NOS2 from the mouse, the growth of bacteria is nearly, you lost control of it. The mice died actually quite fast. So this actually formed this paradigm, this dogma, as people know that T cells making interferon gamma, which further activate infected macrophages that upregulated an enzyme called induced nitric oxide synthesis that produce large amount of nitric oxide and eventually kill the bacteria. So this dogma has been, people have followed it so much, and then so especially guides to the vaccine development, where the current anti-TB vaccine, it's really the target would be induce the strong T cell responses, which produce gamma interferon, and then eventually kill the bacteria. That's the rationale. However, this is what happens. TB vaccine, we have a major failure in the past few years, especially a vaccine candidate that actually can induce very robust T cell interferon gamma responses, but they still fail to induce protective immune responses in human during the clinical trials. And then we thought about it, why the TB vaccine failed, even though there's a very strong gamma interferon responses, and how do we even achieve the sterilization immune responses in the lung? So, and then this is a figure I draw recently, and I think about how to rationale what we've been doing. So this is, I don't know if you people like it, but I try to present it in this way and see if how do you guys like it. So the axis is a time, and then the y-axis is a bacterial burden. So during a natural infection, and initially you will have a very rapid bacterial growth. And then once usually in mice, it will start at 4 weeks, you will have established adaptive immune responses, particularly T cell responses start. So this limiting the bacterial growth. So you have a flat bacteria burden. So it won't go up, but it will maintain there. And then the current TB vaccine is mainly targeting adaptive immunity. So mainly targeting the T cell responses, for example. And then what it does actually can reduce bacterial burden quite a bit. And then actually it moves this, it's the point like a bit of earlier. So you can develop adaptive immune responses, T cell responses a little earlier, faster. So you can reduce the bacterial burden to quite a bit, but it won't eventually clear the infection. So what we think about it to do is whether we can harness the early stage of infection where if there's some cells that can support bacterial growth really well, but it doesn't responding to the gamma interferon during the infection, so it won't clear the infection. So if we harness those cells, we can identify those cells, identify what mechanism actually regulates those permissiveness in those cells. And if we can reprogram those cells at early stage of infection, and so we have vaccine that target innate and adaptive immunity, and where this will eventually create a better strategy that actually may eventually achieve the sterilizations. So this is basically my goals and in the lab. So as you know that macrophages is actually the major host cell types for TB in the lung. It's not only cell types that are infected, but most of the TB lifespan it stays in the macrophages in the lung. And macrophages is during the past 10, 20 years, the research of macrophage biology has been changed quite a bit, and it has been changing dramatically. And in the lung, as well as in other type of tissues, the macrophages actually are having different origins. So this means that precursor cells are very different. And so that actually create a new research area where identify macrophage functions in those different subsets of macrophages with different origins. So this is actually also true in the lung, and we actually benefit quite a bit from this theory during study of TB infections. So in the lung, as many of you know, that in alveolus, we have alveolar macrophages that are actually coming from yolk sac and developing from early embryogenesis in our when we're still in like very small, teeny, tiny cells. And another major type of lung macrophage is called interstitial macrophages. And the differences between these two, besides their location, is different in the lung. And one big difference is their origins are different. So their precursor cells are different. So this interstitial macrophage is coming from monocytes, blood monocytes coming from the bone marrow. And what we find, this is the work when I was a postdoc, we find those two types of macrophages respond to TB infection very differently. And alveolar macrophages coming from embryonic stage, actually, it's a major permissive cell types at early stage of infection. On the other hand, interstitial macrophages are coming from monocytes origins. It's actually restrictive cell types. It actually has better mechanism that regulates, that controls the TB growth intracellularly. So, and it's interesting that major differences of this different type of controlling mechanism between these two cell types is associated with their metabolic status. So alveolar macrophage, as I show here, engage into this pathway called fat acids oxidations into, it's really engaged into mitochondrial metabolism, and which actually can promote the bacterial growth. And on the other hand, interstitial macrophages coming from monocytes and engaging to utilize a large amount of glucose and engage in this pathway called glycolysis and actually respond for controlling of the TB growth. So this actually makes sense because TB is a very unique bacteria. Unlike other bacteria, this bacteria actually really likes fatty acids and cholesterol, so really like a fatty food. So this is a mouse lung infected with TB origin, TB as expressing M-cherry, and the green is the ApoDP, so long chain fatty acids. You can see many of the TB actually accumulates in the macrophages that have a lot of lipids accumulations. And intriguingly, when we harnessing or targeting different cell intrinsic metabolic pathway in vitro, we can actually control the regulate TB growth differently in different. So if we have, this is a bone marrow macrophages in vitro infected with bacteria, but we treated the cells either with 2DG, it's inhibiting glycolysis, or tamoxir, that's fatty acids oxidation inhibitors. We can have either growth, we have enhanced bacterial growth or reduced bacterial growth. And as you can see here, this is a fluorescent image. We can say if you block glycolysis in macrophages, the bacterial growth is almost like tripled, or it's like crazy. When I saw this image, I saw this, I cannot believe the cell can still survive in so many bacteria. And this work actually further confirmed or replicated by Jennifer Phillips' lab at WashU, where they show that also using different setup inhibitor and genetics way that showed fatty acids oxidation is a major pathway that actually promotes macrophage controls bacteria. So this status stage of what we do when I start a lab here in Arkansas, in Little Rock. So our goal is to identify which host phagocytes or that are permissive that can support localized growth of bacteria, even in the presence of a strong protective of adaptive immune responses. And what's the role of cellular metabolism, and how do they regulate those different type of permissiveness in different host cells? And another interesting I want to do is, can we reprogram the cells by targeting different their underlying mechanism that improve, reduce their permissiveness and improve their resistance during TB infection at an early stage. And so this will be, we're trying to do this through targeting cell metabolism and also epigenetic regulations. So I'm gonna share two stories along these two lines, different things. Let's see if people. So hopefully you can that makes sense to some of you guys. So to measure cell metabolism, I think probably most of you know this. Nowadays, this is a popular machine called Seahorse machine that can probe cellular metabolism really well. And this machine actually measures two parameters, the ECAR or extracellular acidification rate, or OCAR as oxygen consumption rate. That's an indication of mitochondrial oxidative phosphorylations. ECAR is an indication of glycolysis activity. And we can actually recapitulate what we find in the Seahorse machine if we purified alveolar macrophages from interstitial macrophages from TB infected mice. We can find alveolar macrophage has a greater oxidative phosphorylations at basal level. And if we use entomoxia treated cells and only alveolar macrophages responding to these inhibitors, however, interstitial macrophages do not responding to these inhibitors, suggesting fatty acids oxidation pathways is active in alveolar macrophages only. This is a great way. However, when I start a lab, first of all, we don't have a cell sorting in the BSL-3. So I was actively looking for the alternative way to measure metabolism using cells without dealing with the cell sorting. And then I also noticed the cell sorting actually has a huge impact on cellular metabolic activities. And based on these two, it's a few papers. This is just two of the representative papers. And then I thought about Seahorse is great. Their alternative way to measure, to probe cell metabolism during infections, and its metabolic measurement can be done at single cell level. Is this even possible? So then I came across this paper published in Cell Metabolism. And it's a very smart way from my perspective. I think I really like this paper. So the way is they link cell metabolism to protein synthesis. So it shows that protein synthesis is one of the major cellular process inside of a cell that actually consume energies. So if you block protein synthesis here, the translation level goes down, and you also have reduced ATP levels. So this process actually coupled together. And then this people, Rafael Aguero, developed these methods using, you can treat your cells from tissues or in vitros using different inhibitors or control cells. In this case, you use 2DG that blocking glycolysis or oligomycin that blocking oxidative phosphorylation, all these two inhibitors together. And you can actually measure the protein translation by flow cytometry, where it's measured incorporation of this antibiotics called puromycin. So puromycin will be incorporated into a newly synthesized peptide, and we have antibodies that against puromycin, so we can measure protein translation activity. And then you can according to the reduction of protein translation inside the cell, responding to different inhibitors, you can actually measure different pathways that actually how much of this pathway contribute to the cell translation, and then it reflects of the activity of that pathways. And then I reached out to the first authors, and we were able to get the kit from him. And then we did experiments in TB infected mice. And then this allowed us to do almost all the major phagocytes in the lung during TB infection. So what we find, this is the different parameters we can calculate. So I just want you to pay attention to the first two. So this blue bar is alveolar macrophages. Its mitochondrial dependency is the highest compared to the other phagocytes. And however, this glycolysis activity, the capacity is the lowest compared to the rest of the cell type, which is very dramatic, very striking. And this is to validate this method, we use a Seahorse to further confirm this method is actually reliable, at least in macrophages. And what we can further using a bacteria expressing fluorescent proteins, we can probe we can probe metabolic metabolism in different cell types using flow cytometry, we can gauge infected cells versus bystander cells. And we can find many of those TB infection in the infected cells, the mitochondrial metabolism is reduced in alveolar macrophages, interstitial macrophages, as well as neutrophils. And then the glycolysis activity is increased. It's increased dramatically in interstitial macrophages and slightly in neutrophils, and also slightly increase in alveolar macrophages. So this is further confirmed by the RNA sequencing data, where this is a glycolysis pathway. If you have infected macrophage, alveolar macrophage, you have a slightly increase of glycolysis, but this increasing is amplified significantly more in interstitial macrophages. And so this method is pretty reliable. It's consistent, can validate by seahorse and sequencing data. So what we actually attract us is this population called neutrophils. And the reason this interesting to us was because neutrophil is a cell type that actually people know traditionally is really rely on glycolysis. However, during TB infection, it has many mitochondrial dependency. And then we dig out into literature and we find that neutrophils really are much less studies compared to other type of cells in tuberculosis. And many of this data is controversial. Again, this cell is difficult to study because it lives short. It's a very short lifespan, not easy to work with, hard to culture those. And interestingly, the susceptibility of TB to TB, it's highly associated with numbers of neutrophils. That means if you have more neutrophils in the lung, the bacterial burden is much higher, and you have a worse infection outcome. And again, people thought neutrophils really solely rely on glycolysis, but we find in the TB infection, it doesn't. It's not the case. So then we start with some simple experiments where we just compared bone marrow neutrophils compared to the bone marrow macrophages, and we infected with TB and put on the seahorse at a very early stage of infection, and we find how do they respond metabolically against the infection. So at neutrophil, so as you can see here, neutrophil infected cell, infected neutrophil enhance their glycolysis activity dramatically, but their mitochondrial activity doesn't change much. This is opposite in macrophages, where mitochondrial activity actually decrease in the initial infection, but the glycolysis activity doesn't change much. So again, this is in vitro. So we try to recapitulate what's differences between in vivo and in vitro. So we perform this single cell sequencing by purifying TB infected lung phagocytes from mouse, and we find from neutrophil populations in mouse, we find 3 different small clusters from TB infected mouse lung. And this is just a marker that highly express on neutrophils, and just showing all the cells are actually neutrophil populations. And we find there's 3 subsets of neutrophil, especially this neutrophil population 2. There's many genes that are differentially regulated, including amino acid class 2, INOS, and HIF1 alpha. So that's what attracted us. Okay, that's population so unique. So this is a synchronous. We find in that population that highly upregulated this Fc gamma receptor or CD64 in that population. And again, this is just a Valin plot to show they're also highly upregulated class 2 and INOS. And then we further trying to look at those cells by flow cytometry. We can find in the infected mouse lung, there's a subpopulation of neutrophils that are significant upregulate CD64 positive, and we can identify those 2 populations. And then this flow cytometry also confirms highly expressed of class 2 and a slightly increasing amount of INOS in the CD64 positive populations. And then we're curious, what does the cell do? So before that, we also want to try to find those type of cells in the monkey model, because the reason is that people always argue mouse model is not great for studying TB. It's so different from humans, and monkey, so non-human primates, it's a way better model, and it's almost recapitulate fully of what infections in human. So we download the data sets from this recently published paper from Alex Schlick's lab and Sarah Fulton and from Harvard. So they did this crazy single cell sequencing in lung granuloma from monkeys infected with TB. So we extract the neutrophils and we actually find 4 neutrophil subsets. And surprisingly, this neutrophil subclass, it's neutrophil 3 population in the monkeys express many genes that are similarly to what we find in mice, that neutrophil that express CD64. There's actually nearly half those genes we can identify in this mouse population, neutrophil 2. So suggesting this, we may be finding one type of neutrophil that actually exists in 2 different diseases, 2 different models of the TB infection. And then we reach out to Josh Matila's lab, who actually has a large monkey TB facilities at Pittsburgh. And he has many of those monkey lung granuloma. This is what it looked like in monkey lungs. And then he actually did staining of neutrophils using CD64. So he actually found in granuloma of neutrophil in granuloma of monkey lungs. So their CD64 level expression percentage and mean of fluorescence intensity also increase as well, suggesting these cells indeed and may be present in the lung granuloma of TB infected monkeys. So this is very intriguing to us. So it promote us to do more in functional analysis. So one thing we're always interested in is the metabolism of the cells. And so of course, the first thing we look at is cell metabolic activities between these 2 types of neutrophils. So we find, again, those translation level in CD64 positive cells indicated by puromycin uptake, it's significant highly. And also this cell, it has a higher increase in mitochondrial dependency. And again, this mitotracker staining indicating the mitochondrial potential is also increased in this cell population, as well as this neutral lipids level. So this cell, CD64 positive cell, is highly metabolically active compared to its the counterparts of CD64 negative neutrophils and metabolically. And this is further supported by, if we do the single cell analysis, and we can find glycolysis genes and oxidative phosphorylation signature genes in the single cell plots also upregulates in these populations. And we further confirm this by doing the seahorse analysis, confirm that this population has both high ECAR and high OCAR, indicating they're highly active in both glycolysis as well as oxygen consumption in mitochondria. And then the next question we try to address here would be, this cell population didn't exist in the naive mouse lung, so it's actually driven by an infection. So we're wondering what drives, what signal or cytokines promotes the generation of this new populations. So we start with in vitro experiments when we culture with TB or interferon gamma. We actually tried a bunch of different cytokines. So this is one only shows differences. That gamma interferon, so let's just say here, if you have TB, the CD64 expression level goes up, and it's an additional gamma here actually promotes further upregulation of CD64. The same with this neutral lipids accumulation in the lung. So suggesting gamma interferon actually is responsible for promoting to generate this new CD64 positive neutrophil population in the lung during TB infection. And this is also confirmed by the in vivo data. If you infected gamma knockout mice, you have the neutrophil numbers stays the same, but you have less CD64 positive neutrophils in the lung. So what does this population do, right? So this is actually the question that we care the most. So I think we need to, so to probe this, we actually, sorry, I'm just trying to try to play this movie if I can. Excuse me, here we go. So to probe the function of the cells, we actually use this reporter. We actually get it from my good friend, Shuming Tang. Something wrong with the movie, I'm sorry about this. But again, this reporter strain is probe the bacterial replications. So as you can see here, the red bacteria consistently express M. cherry. And those bacteria actually start replicating, will have this GFP foci. This is a fusion protein indicating of single strand binding protein. So for example, this bacteria inside of cells is replicating actively, but this bacteria at this moment is not replicating actively. So we find there's no differences in terms of bacterial replication activities. Remember, the CD6 populations, positive populations, expressing INOS, which is known to be protective, part of the protective immune responses against TB, but apparently in these cell type populations, bacteria doesn't care. And unfortunately, even in the presence of gamma in those populations, we can actually purify, there's more viable live bacteria from these populations, suggesting this population is very permissive, highly permissive to the infection during in the lung. So, and we're wondering what actually caused these differences of permissiveness during infection. So again, if we measure fatty acids uptake by flow cytometry in vivo, this population uptake more long chain fatty acids. And this is corresponding to the highly upregulated receptor of fatty acids. This is CD36, is uniquely upregulates on this newly identified population. And accordingly, there are two papers that are actually supporting the lipids metabolism both in cancer, published. One is in Nature last year, one is in Nature Communication a few years ago, and said neutrophil actually engage in mitochondria and fatty acids utilization during cancer. So again, seems like this is also the case in TB infection. So, and then my question is, if we can target in that pathway, can we reduce the bacterial growth? So to do that, we infected CD36 knock-on mice. So in those mice, the CD36 cannot, it's not functional. And again, in neutrophils, the fatty acids uptake is reduced significantly in those mice. And again, if you look at the bacterial fluorescence or the number of bacteria actually can recover from those cells, only in the CD64 positive neutrophils, you have reduced bacterial numbers, suggesting if we're targeting this pathway, that fatty acids uptake pathway, you can actually reduce bacterial growth, can compromise this specifically in these populations. So that's what we have done in that part. We identified, I think this is a new population of neutrophils in the lung that actually is responsible for early stage of infection that function as a permissive cell types. And the mechanism, as we believe, is through the fatty acids uptake and enhanced mitochondrial metabolism. So what I think is important to show here is cell metabolism actually regulates immune pressures inside of cells for the intracellular bacteria, and also regulates the nutrient availability. So right now, if only targeting enhanced immune pressures, the bacteria may be okay. They may survive fine because they still have enough nutrients inside of the cell. So in the future, we may have to consider pathway or drugs that more targeting the nutrient availability. Combine this, we probably achieve bacterial control better at early stage. So, and then I'm gonna switch gears to talking about something that we've been doing quite a bit in the lab. Which is how to reprogram innate cells during infection at early stage. And so this work is mainly done by this very talented, this is my technician. So he was freshly graduated from the college almost 2 years ago. So being in my lab almost 2 years. So he did all this work. So I'm very lucky to have him, Andrew Tuckers. So before that, I want to introduce this concept called pre-immunity. So this is a way that measures that's newly emerging area, has been very popular in the field, in many different fields. So this is describing memory response in innate cells. So as many of you know, traditional people think only adaptive like T cell, B cell can generate innate memory. And this has been challenged in the past few years, past decades, I guess. So innate cells, particularly macrophages and monocytes, also can have this memory-like responses, and it's called trained immunity. And this trained immunity is largely regulates by this epigenetic reprogramming and metabolic reprogramming in the cells. And this has been showed being involved can induce protective against TB and also have protective responses against tumor. And of course, at the beginning of the COVID pandemic, people actually spent quite efforts to try to use this BCG vaccine to induce trained immunity to show whether it can induce protect from COVID infection. So what our question was, can this trained immunity be induced at this specific mucosal site? In our case, it's the lung. Can we induce trained immunity directly in the lung? And if so, what intrinsic factors actually can regulate the trained immune responses? So to do that, we use this chemicals, beta glucan. So it's the reason to use that is because it's one of the most potent trained immunity inducers that people have been used. And the other thing is it's relatively safe. So if you look at NCI clinical trials, they have over 100 clinical trials using beta glucans in humans. So it's relatively safe for us. It will be easier to translate in the future. So this model is quite simple. So we gave mouse beta glucan intranasally. So apply beta glucan directly to their lungs. And after 2 weeks, we find there's really no differences in any of this major cell population. No, the numbers fine. We don't have any differences. But interestingly, we find after 2 weeks, the activation markers of lung alveolar macrophages is significantly upregulated, including CD80, CD86, CD206. That's indicating of phagocytosis, and also their mitochondrial potential is also enhanced after beta glucan treatment. And if you do the seahorse analysis, you can find that's enhanced oxygen consumption after the FCCP treatment. And also there's an increased glycolysis activity inside of cells. And this is not because of beta glucan induce any kind of inflammations. And if we measured like 32 cytokines in the lung bell, but we didn't find any major differences. This is only cell that the cytokine that actually detected, and TNF alpha, interferon gamma, and the rest of many of those we couldn't even detect any of those cytokines in the bell. So whatever has happened to the alveolar macrophage activations after 2 weeks is not because any inflammation is still present in the lung. So using like hand-dimensional flow cytometry analysis, we find this is a difference. We find in the alveolar macrophage populations, there's a population emerging compared to the control mice. This is two markers, cyclic F, CVMC, that's the markers highly expressed in mouse alveolar macrophages. And you can see this statistics is very consistent every 2 weeks post beta glucan treatment. And this is just markers expressing on this two type of alveolar macrophages. One difference is CD11B, it's a myeloid marker. It's an integrin highly upregulates on that population that are cyclic F low. And also it has increasing expression of class 2, indicating this cell is activated. So we, based on the increasing of CD11B markers, we were wondering whether these populations are coming from monocytes after beta glucan treatment. So we use a CCR2 knockout mice where the monocytes is nearly abolished. So we find this population is disappeared, indicating this generation of this new population after beta glucan treatments is dependent on the CCR2 receptor, and so it's dependent on monocytes most likely. So we then utilize to confirm this discovery. We use a monocyte fit mapping mice. This is a CF3CL1 CRE-ER. We cross it to TD tomato mice. So all the monocytes in these mice after treat tamoxifen will become red. So we can follow their phenotypes in the long term. And this is exactly what we find. We find beta glucan treatment induce a generation of TD tomato positive alveolar macrophages, suggesting this population is coming from monocytes. This is confirmed that only cyclic low population have nearly 70% of those cells that are actually monocyte derived, suggesting beta-glucan actually promotes accumulation of monocyte-derived alveolar macrophages in the airway. And interestingly, this generation of this population is dependent on interferon gamma, but it's not dependent on T cell or B cell adaptive immune responses. So suggesting some innate cell populations producing gamma interferon is responsible to generate these populations after beta glucan treatment. At this point, we don't know what cells that are actually doing this. We have no idea. And then, so to further delineate the mechanism, what does beta glucan do to macrophages? So we start with very simple model. We inject beta glucan to the mice IP, and then we purify the bone marrow and culture bone marrow macrophages, and then we perform the proteomics analysis here. And this is the path we find. There's many metabolic pathways that are dysregulated after beta glucan treatment in the bone marrow macrophages, especially this mitochondrial pathway, including oxidase phosphorylation pathway, mitochondrial translation, and fatty acids beta oxidation are reduced dramatically in the beta glucan treated cells. So this allowed us to further confirm whether this monocyte-derived alveolar macrophages induced by beta glucan is metabolically different from its compartment, whereas embryonic derived alveolar macrophages. So again, the activation markers on the TD tomato positive monocyte-derived alveolar macrophages is highly upregulated, except this one, SERP1 alpha, is negatively. This one has been shown is negatively associated with activity of alveolar macrophages. So this is a reversed. So this actually confirmed, indicating this population is highly activated. And then we purify those two populations and put on seahorse, and we find TD tomato positive monocyte-derived alveolar macrophages are highly glycolytic, but their mitochondrial activity is reduced compared to the TD tomato negative embryonic derived alveolar macrophages. And remember, just both populations, they're in the same environment. They're both in the airway. The only differences is their origin. So one is coming from embryonic, one is from monocyte, but their metabolic and their activation is so different. And then this is we further discovered that the monocyte-derived alveolar macrophages have reduced lipid uptake and also have reduced neutron lipids or lipid droplets inside of those cells. And so that's suggesting it has reduced lipid metabolism. This is correlates the reduction of mitochondrial metabolism. So to measure their function, so macrophage has it's very known to be able to phagocytose very actively and also produce different type of cytokines. So we measure their functions by doing this very standard phagocytosis assays. So we gave the Ferrando labeled E. coli. So this is a fluorescent bacteria when it's sensing the lower pH inside of phagosomes. So we find this TD tomato positive alveolar macrophages has enhanced dramatically of their phagocytosis activity compared to the TD tomato negative. And again, on the cytokine production, if you stimulate this different type of cells with LPS, so in the non-stimulated cells, you don't have much TNF production. There's a slightly increase after the TD tomato negative cells are seeing LPS. However, the TD tomato positive cells produce huge amount of LPS, a huge amount of TNF alpha after stimulated LPS, and this TNF production is dependent on glycolysis, not on oxidase phosphorylation. If you give them inhibitors, so suggesting their active glycolysis pathway, at least it's involved in promoting the cytokine productions once they see the bacteria or infections. So to further delineate their functions, so we in collaborating with one of the Lingxi Li, so with the faculties in our department, we performed the single cell ATAC-seq where we purified monocytes from the lung from PBS treated or beta glucan treated mice, and this is what we find of their chromatin structures. So the blue is beta glucan, it's a PBS control group, and the beta glucan treated cells are red. So this population is uniquely induced by beta glucan treatment. And after characterizations, those are the monocyte-derived alveolar macrophages. So this allowed us to compare the chromatin structures into this different type of populations. So again, we're still processing. We just got this data recently. We're still processing, but there's some some interesting things already shown. So if you see here, these are the two populations of alveolar macrophages that are both from embryonic derived in PBS or beta glucan treated cells. They really don't have much differences, suggesting embryonic derived alveolar macrophages, they don't respond to beta glucan treatment really well. They're pretty quiescent in the lung. However, if you see beta glucan treat this cell population from embryonic derived versus monocyte derived alveolar macrophages, they both say beta glucan, but there are so many differences, chromatin alterations between these two populations. And again, the only differences between these two populations is their origins. One is from embryonic, one is from monocytes. So we are still processing. We haven't finished an analysis, so we don't. I don't know the exactly pathway that has been altered. But we did a transcription factor enrichment analysis to identify what transcription factor may be involved in regulating the differences between these populations. Here, this is a few things we find. There are many, the PPAR families is well known to involve in lipids or catabolism or lipids metabolism in macrophages. These are highly enriched in embryonic derived alveolar macrophages, along with many of these nuclear receptor, and I have no idea what do they do. So if anyone here knows, I'm happy to chat more about this. And on the other hand, in the monocyte-derived alveolar macrophages, we find many of these factors, a transcription factor involved and regulates that inflammation responses. It actually supports the data that actually show that this population is very good at mounting inflammatory responses during infection. So the last piece of data we have is we want to treat TB. That doesn't even matter, right? That is beta glucan intranasal treatment even matter. So this is what experiments we did. We treat the mice with beta glucan intranasally, and we wait for 14 days, and we challenge with Mycobacterium tuberculosis. And so this is the thing we like to see, that we have almost 1 log, or I don't know, 3/4 of logs reduction of bacterial burdens after we treat beta glucan in the lung. And this is alveolar macrophages, they are highly upregulated of class 2. They're more activated compared to the PBS group, and their INOS level is also increased. So this is great. So with thinking, if we give beta glucan, we can actually reprogram alveolar macrophages by inducing different ontogeny, alter their ontogeny. So we may be able to protect bacterial infection early stage better. And lastly, if we purify the 3 types of macrophages from control group or beta glucan treated, TD tomato positive or negative. So one is embryonic, one is monocyte derived. We challenge with TB ex vivo, and we find only the beta glucan trained TD tomato positive monocyte derived alveolar macrophages can produce large amount of this pro-inflammatory cytokine. Many of those are being reported to be associated with protections during TB infections. So overall, this is the model we try to put together, where you really need to train the trainable cells. So alveolar macrophages from embryonic stage really not good at being trained by beta glucan. Then they don't mount much strong trained immunity. And so to be able to induce a trained immunity, you will have to train monocyte-derived alveolar macrophages. So the ontogeny actually here is important. So I think this is everything that I want to show here. I'm gonna stop here, and I just want to acknowledge we're in a new lab. I'm a new faculty, so we don't have a big lab. So again, the second part of work mainly done by Tucker Andrews, the very talented technician, so who is going to the medical school this year. So Ashley is a new tech, just started recently, and Fisher is my first technician. Helped me set up the lab and did some works in neutrophils initially. And again, they have a lot of collaborators at different locations. And I just want to highlight Lin-Shi Li. So she's a T-cell biologist in our department who helped us do the single-cell ATAC sequencing. and Rafael Aguero from France to give us the science kit so we can do the single cell metabolism analysis. And Josh Matila, who actually helped us do the monkey study, and Shuming Tang at Tufts University, who provides us the TB reporter string, so we can do all those imaging analysis. And also want to appreciate our funding resource from our COBRI Center through NIGMS, also American Lung Association for supporting our research. So I'm going to stop here, and if I'm happy to answer any questions from you guys. Thank you so much for your attention.